So for today, we will start our online class with the chapter 1 lah, biological molecule. Okay, so as you guys know that, but chapter 1 we need to learn 5 biological molecule: water molecule, carbohydrate, lipid, protein, and also nucleic acid. So these 5 molecule actually you are totally learn in SPM. So in this chapter I only can say it's just a revision chapter lah to you guys. So hopefully everyone can clear about my class. So if any question, you just type in the chat box lah. Okay, so firstly we need to enter water. Okay, so what is water? So I think I don't need to explain, right? Anyone know what is water? As you guys know your knowledge from your kindergarten, everyone know what is water, and we know that water is very important. So this chapter you need to know what is the structure of the water. So we know that this is the structure of water molecule that you learn in chemistry. So we know that the chemistry, the chemical structure, the chemical formula of the water is H2O, right? H2O. So why is H2O? Because it's containing 2 hydrogen atom and 1 oxygen atom, right? So this is the basic chemistry. So we know that we have 2 hydrogen atom, and we know that hydrogen atom is partially positively charged, and oxygen atom is negatively charged atom. Right. If you still remember that you learned about the covalent bond, formation between the oxygen atom and also hydrogen atom, because both atoms are non-metal, right? So they will form covalent bond. So we know that oxygen containing 6 electronic configuration, right? The electronic configuration is containing 6 electron, and hydrogen is containing 1 electron only. So So the oxygen and hydrogen will share 1 electron to each other. They will share 1 electron to each other to form H2O. So we know that oxygen atom is slightly negatively charged and hydrogen atom is slightly positively charged. And we know that water is polar molecule. This is the polar molecule. Why? Because it is a polar molecule, because it has unequal distribution of electrical charge, because the charge between the oxygen and hydrogen are different. The charge between the oxygen and hydrogen are different. Oxygen negative, hydrogen positive. And hydrogen atom, the water molecule, is formed the band tetrahedral shape. This is the band tetrahedral shape, meaning that it containing 2 bond pair electrons and 2 lone pair electrons. So you know, if you see this diagram, 2 bond pair meaning at here. We know that oxygen containing the 6 electron at the outside electronic configuration. They are containing 6 electron, and 2 of the electron will be used for covalent bond formation, and another 4 electron will be known as lone pair electron. And I need to tell you guys, this one is that later you will learn about chemistry. And later your Sem 1 chemistry in chapter 3 chemical bonding, you will learn back the chemical bonding. You will know that why water molecule is forming band tetrahedral shape. They are the theory, but this is not chemistry class. So in biology, you just need to know water molecule, the water atom, the chemical structure of the water molecule is band tetrahedral shape. Why? Because it containing 2 bond pair electron and also 2 lone pair electron. Lone pair electron is at the outside one, and this one will form the band tetrahedral shape. So due to all this reason, we consider as water is polar molecule. Polar molecule. Okay, and then water molecule is very special molecule because it can form hydrogen bond. compared to others molecule, if they are the interaction, if you still remember that you learned in SPM chemistry, if you got any interaction between the molecule, which is a non-metal molecule, with the non-metal molecule, they will form either covalent bond or either form van der Waals force, right? Normally is van der Waals force, weak van der Waals force. But hydrogen bond, water molecule is very special. When the water molecule are interacting to each other, they will form short bond. The one of the bond we call as hydrogen bond. So this is the bond that form between the oxygen and also hydrogen too. But the bond formation is between the water molecule. You need to differentiate between covalent bond and also hydrogen bond. Covalent bond is that the bond form the water molecule. One water molecule structure, they containing two hydrogen bond. And then hydrogen bond is that the bond formed between the water molecule. The water molecule are different. So this is the first water molecule and this one is another water molecule. So they can form hydrogen bond between hydrogen atom and also oxygen atom. Why? Because of the unequal charge. So visual ordinary water molecule with opposite partial charge were attracted to each other, but this one will not form covalent bond. They will form the weak bond, the quite weak bond, which we call as hydrogen bond. So this hydrogen bond is weaker compared to covalent bond, but stronger to van der Waals force. This is at the intermediate lah, So the positively charged hydrogen atom of one water molecule are attracted to the negatively charged of another water molecule, normally is nearby, will call hydrogen bond. So you need to know how to draw it. So we know that one water molecule can form 4 hydrogen bonds to each other. So you see this diagram. This is a central one. I use the central as example. We know that they're containing 3 atoms, right? One water molecule contains 3 atoms, 2 hydrogen atoms and also 1 oxygen atom. So one oxygen atom can form 1 hydrogen bond to another water molecule, same to here. So there are 2 hydrogen atoms, so they already conform 2 hydrogen bond to each other. So 1 hydrogen atom form hydrogen bond to another water, and another hydrogen atom form another hydrogen bond to another water. So 1 oxygen can form 2 hydrogen bond. They can form 2 hydrogen bond. Why? Because oxygen containing 2 lone pair. They have 2 lone pair electron at there. So that is why that hydrogen bond can form for oxygen atom is 2, not 1. So you need to how to draw it. You need to draw like this, but in 2D structure, not 3D. This one is 3D structure, right? This will in 2D structure. This one that you need to how to draw it. And also you need to according the shape of water molecule, not like the 90 degree. You need to follow this one. The water molecule that you need to draw, you need to follow this. The angle is 104.5 degree. This is also you need to memorize it. Okay, so this is the spatial, and this hydrogen bond characteristic is very important because this one is contribute to the physical properties of the water molecule. This one, all these hydrogen bond, structural, chemical structure, all are the chemical property of the water. And this hydrogen bonding is very important that contribute to the physical property of the water molecule. Okay, now we go to the main water property. So there are 6 main water property that you need to know. So first property, everyone know, water is a universal solvent. This one is one of the chemical property, and others are physical properties. Low viscosity, high specific heat capacity, high latent heat of vaporization, high surface tension, and has maximum density. These are all the physical properties. Only the first one, universal solvent, is considered as chemical property. Why you need to differentiate them? Maybe in exam, the question only want you to talk about physical property only. If talk about physical property, this one universal solvent you don't need to write it. If you write it, no marks. Because the question is talk about want you to write about physical, not chemical. So you cannot write universal solvent lah. You write it, no marks lah, no mark given because you are not answering the question. Okay, so we will go through it one by one. So as we know that why we call as water is universal solvent, because you compare to other type of liquid called like alcohol, acid or salt solution or other and any type of water liquid molecule, water can dissolve almost all substance, especially it can dissolve the polar substance, polar solid substance. So for example, it's salt lah. We know that salt, some main salt like NaCl, sodium chloride salt can dissolve in water, but not all salt can dissolve in water. This one I think you already learned in STPM. Some salt can dissolve in water, some salt cannot dissolve in water. But in biology, normally the salt that we will use is NaCl. Because NaCl is the most common table salt that our human and also any organism can be used. Need to take one. So we will, in general biology, we will use the sodium chloride as example. So normally it is table salt lah. We will call about table salt is about sodium chloride, only sodium chloride. So we know that sodium chloride can dissolve in water. How the sodium chloride can dissolve in water? So this is the chemical property, the properties. So we know that salt is solid. It is solid particle. So it will arrange at like this one. This diagram. This is a solid particle of the salt that arranged in solid form. So sodium chloride are tightly arranged to each other. And once it we enter into the water, dissolve in the water, the reaction is happening where the sodium ion, the sodium ion will be attracted to the negative feature of oxygen molecule, negative feature of the water, which is oxygen. The oxygen will attract the sodium ion toward itself because of oxygen. Meaning that they will have the interaction between the oxygen and also sodium ion lah, but they will not form, just attract only. Dia macam tarik itu sodium daripada sini ke sana, macam tarik je. Similar to chloride ion also can be attracted by the hydrogen region, which is a positive region of the water molecule. So it will attract the chloride ion from the salt, from the solid salt. So that is why the table salt can dissolve in the water, due to this attraction. So you will not see any solid particle in the solution when the solid salt is already entered into the water, because they already interrupt the water molecule, can affect the solid structure of the salt. So that is why that we call as universal solvent. So not only table salt, if you apply in the biological system normally is water soluble protein that can soluble in water. The water molecule also will have the same interaction happening inside there. So normally like the human lysozyme, the enzyme also can be dissolved in water, so they will have the interaction between the protein molecule and also water molecule. So yeah, so water will act as the universal solvent. Okay, next is about the physical property of the water molecule, which water has low viscosity. So what is the meaning of viscosity? Viscosity is that the weight of the liquid resist to the pressure, or resist to friction, or resist to the force, all that. In bahasa Melayu, if not mistaken, if high viscosity meaning dia sangat dekat, dekat sana, dia tak akan bergerak punya. Macam oil lah, if you consider to oil has high viscosity, but water has low viscosity, meaning that the flow of the water is very smooth. Dia tak akan dekat satu benda punya. Satu benda punya. Dia memang smoothly pass through one. So why water has low viscosity? Make it low viscosity. One of the reason, hydrogen bond. Hydrogen bond, although it is stronger than van der Waals force, but it is also considered as weak. Compared to the covalent bond, compared to the ionic bond, it is also the weak. It is very easy to break down. Also very easy to break down. When the energy is quite high, it's quite high only, then the hydrogen bond is will break down. And water can slide easily to each other, meaning that the interaction between the water and water molecule are not very strong one. It can, for example, this water molecule will not have the hydrogen bond interaction like this anytime. Maybe it will have the interaction bila satu jam lepas dia pergi cari water molecule yang lain. They will find other water molecule to form other hydrogen bond. So the interaction will not retain long, retain only short only. Retain only short. So that is why it can slide easily to each other, and it is act as the medium of transportation in living organisms. So due to this low viscosity, it will become the main component to transport all the component within the organism, like our blood. We know that blood not only containing erythrocyte, not only containing leukocyte, the major component of the blood is water. Why? Because it need to transport. If no water molecule, if the blood only containing erythrocyte, leukocyte, only the cells, then this cell actually cannot transport one. Dia tak boleh bergerak punya. It can move due to the water. Water memang satu medium, macam highway atau macam not highway, macam kereta untuk kepada sel untuk erythrocyte dan juga leukocyte. It's like a transport, like the transport car for the cells that can travel along the blood vessel. Not only cells, and also including the nutrient, any nutrient like the salt, and also organic nutrient. All these are be able to be transported inside the water, and also it act as a good lubricant to reduce friction within body. So this is the explanation about the low viscosity lah. Another one, this one is, I think you already also learned in SPM. Water has high specific heat capacity. Okay, first you need to know what is the meaning of specific heat capacity. The definition of specific heat capacity. So normally in general, I just want to tell you in general, it is the amount of the heat that can cause 1 gram of water to increase 1 degree Celsius. Meaning that I need to use how many heat, the amount of the heat, the energy, how much of the energy to increase 1 gram of to increase 1 degree Celsius in 1 gram of water. So that is why that you can see this unit 4.3 joule. This is the energy, meaning that we need to use 4.2 of the energy to increase 1 degree Celsius of water molecule in 1 gram of water molecule. Awak nak naik suhu dia, if you want to increase 1 degree Celsius of the water molecule in 1 gram of water, you need to have 4.2 joule of energy. So this one, if you compare to others molecule like alcohol or alkene or other organic solvent, you will see that this is very high. It's quite high already. It's already considered high. Normally in alcohol, if you compare to alcohol not even only 1 only or less than 1. Some of the alcohol less than 1. So that is why the alcohol is very easily to evaporate. You know that you wash hand. After wash hand, you use the sanitizer. When you spray it to your hand, later it will quickly evaporate. The liquid is quickly evaporate. So the water for the alcohol, the evaporation, the specific heat capacity of the alcohol is very low. But you compare to water, if you want to increase the temperature of the water molecule, very difficult. Even though you use the gas or electrical gas or any gas, you need to get the water boil you need to also take 5 minutes, 10 minutes, right? 5 minutes, 10 minutes, right? So yeah, so we consider as the specific heat capacity of water is very high. Very high. And this one will, what are the importance? Why you say high? Kenapa dia kena high? What are the importance toward the biological life? So meaning that the water temperature in the cell will remain, the environment of the cell is constant. So if no water molecule inside our body, meaning that when the weather is very hot, your body, your cells will die, because your cell will also, your body temperature will also increase. Like when the weather is like 40 degrees Celsius, your body temperature will also increase up to 40 degrees Celsius if you do not have water. So your cell will die lah. Akan mati juga lah. Awak tak boleh tahan kan, sel pun tak boleh tahan, akan mati juga. So water can make your body temperature remain constant. Your internal environment is remain constant. So similar to ocean, like your fish. So if you know that when you are very hot, you will go to, akan pergi pantai kan, atau is very hot weather, you go to swim. Why you need to go to swim? Because at very hot weather, the water in the swimming pool or the water in the sea is low. For you is low. Why? Because it had a high specific heat capacity. You need to increase the temperature of water is very difficult. So that is why that if you due to this property, so that the ocean temperature can be maintained. The ocean organism, the marine organism can be survived. If does not have this kind of property, meaning that if the surrounding temperature, the environment temperature is very high, your ocean also very high. And then maybe all the fish, all the jellyfish, all the seafood semua tu akan mati lah. Memang mati. And also enzyme, biological function. It ensure the biological function. So yeah, okay. When we talk about specific heat capacity, we will also talk about the heat vaporization. Okay, so both of them is sibling. So specific heat latent heat vaporization, I think this one you also learn in SPM 2. So this one is that if you want to increase, if you want to make the definition, you want to how much of the energy is needed to make one molecule transfer from one phase to another phase. Another word, you apply water meaning that how much of energy I need to use to transfer liquid water molecule into gas. We know that gas is a form of the water molecule, right? If you boil the water, you will have many gas, right? So that gas is the gas form. Gas form is another phase, right? So if you want to want to make the water become the gas, you need also energy. So due to specific capacity is very high, automatically your latent heat vaporization of water also very high. So this is one is you need to 580 calories lah, to increase 1 gram of water, to make 1 gram of water break down the hydrogen bond, so that the water molecule can vaporize. So this one is very important in cooling effect. This one provide the cooling effect to you. Why we say that it provide cooling effect? We know that when in very hot temperature, you are sweating, right? Awak akan berpeluh. Memang awak akan berpeluh. Kenapa awak nak berpeluh? Because they need to reduce your body temperature, reduce your surface body temperature. Or the most easier thing, when you are very hot, sangat panas, awak akan pergi mandi kan? You will go to washroom to take a bath. Why? To cool up, to make you cool, lower your temperature. So why water can do that? Why water can provide the cooling effect? Due to this. So the main important is when there are water molecule on in your skin, like this elephant lah, they flash water to their head, to their skin. When there are water molecule remain on the skin, meaning that the heat, the water molecule will absorb the heat first, rather than your skin. If no water molecule, meaning that your skin will absorb the heat. But if there are the water molecule at there, they will absorb heat first, so that it protect your skin. And another thing, due to the water have the very high heat vaporization rate, so that water is not easily to evaporize one. You want to vaporize the water molecule from your skin, is not easy. So that it provide cooling effect. Not easy, so it provide cooling effect. Another one is that water molecule with the greatest kinetic energy live as a gas, making the remaining water cooler. You see this water molecule with the greatest kinetic energy live as gas, making the remaining liquid water cooler. So you know that water molecule is arranged in the liquid from the hydrogen bonding. So when they already evaporate, they will provide more cooler to the lower part of the water molecule. So this is one of the property lah. Another one is high surface tension. Another word is you can see there are the insect can walk on the water one. Mereka boleh jalan dekat air punya. Human tak boleh lah. Only insect only can lah. The smaller kind of organism can, but human tak boleh lah. Because the body weight of human is very high. But for the smaller organisms like insect, they can. Why they can do this? The another term is water has high surface tension. So why this? High surface tension is apply is walking on water. So due to one of the force is cohesion force. Meaning that there are the strong hydrogen bond of water molecule bound to each other, especially at the layer of the water molecule. If you see this diagram, at the layer of the water molecule, the hydrogen bond is very strong compared to the lower one. The water molecule will be arranged very badly at the upper layer compared to the lower layer. Lower layer. The lower layer one, the water molecule can move freely one. Dia boleh bergerak sampai. Tapi yang upper layer punya, dia tak akan bergerak. But no movement. Have move. Ada movement tapi kurang. They will badly to each other. This one contribute surface tension. So this one will provide a strong layer on the surface, make the water is very hard to break on the water surface. Ini memang, if you try go to water molecule, go to swimming pool, you know that awak nak tembak tu water punya, awak nak pukul water molecule water, awak pukul je, awak rasa sakit. Memang awak rasa sakit punya kan. If you have go to the swimming pool, want to play the water, awak ambil air atau awak pukul air, memang awak tangan punya sakit punya. Due to this lah, surface tension. Because it make the water molecule is very hard to break. So for the insect, they will feel like water like a lantai lah, macam lantai kami. So that is why they can walk on the water. So on the surface, water has greater attraction for each other. So this one will contribute high surface tension. Another one is high surface tension will also contribute another important application is cohesion adhesion theory, which is the transport in blood. This one is very important too. Later you will learn in semester 2. But before you enter semester 2, I think you also learn in STPM, transport in plant. How the water molecule is transport from the root to the leaf using transpiration pool, right? And inside the leaf they have two force, right? Cohesion force and also adhesion force. We know that cohesion force is Cohesion force is the interaction between water molecule, and adhesion force is the interaction between water molecule and also the wall of the xylem vessel. This one I just recap. Although you are not entering semester 2 yet, but I assume that you already learned in SPM. So if you already forget it, never mind, you don't need to focus it. But in semester 2, I remind you, you will learn back again. But this one is just I need to talk about the application. High surface tension will contribute this. So that is why that water molecule can pull up against the gravitation. You know that Earth containing gravitation, meaning that if logically gravitation will cause the water molecule akan jatuh punya, will cause the molecule fall down, not upward. This one is already against the gravitational force. Why like this? Due to the hydrogen bond. The most important keyword is hydrogen bond. If no hydrogen bond, if no this hydrogen bond, then no surface tension and no transport like this. Due to the presence of hydrogen bond. The keyword. Very important. So yeah, so this one. And final one, if no mistake, final one, water molecule has the maximum density at 4 degrees Celsius. This is very special compared to other type of the molecule component. Others, we will know that the 0 degrees Celsius is the freezing point, right? We know that 0 is the freezing point of the water molecule. But freezing point not indicate that the water at the ice is the solid form of the water, right? Ice has the maximum density. No, ice does not have the maximum density. Ice, if you want to make ice, your temperature must be lower than 0, right? Normally it's -10, -8, -9, right? In your freezing. So, but ice does not have maximum density. Why? Due to the arrangement of the water molecule particle is different compared to other type of solid particle. If you still remember in SPM, that chemistry that you learn about the solid particle, the arrangement is normally is tightly to each other, right? You write a circle, macam circle tu semua tightly to each other one. And liquid macam loosely. And then gas sangat kecil, sangat kurang. The molecule is very rare. The atom is very rare. But in water, you cannot draw like that. You can draw like that due to the hydrogen bond also. Why the water has the maximum density at 4, not 0? Due to the hydrogen bond. Because when the hydrogen bond, it will decrease the temperature. Of course, the kinetic of the atom, it will be decreased. They will stop moving. But due to the structure of water molecule is very special, and also it can form hydrogen bond. They will arrange in the crystalline structure form like this. You see that one water molecule, they will form 4 hydrogen bond, right? 1, 2, and then 3. This one hydrogen 1, and then this one 1, and then 2 hydrogen from another 2. So they will form 4 hydrogen bond. And once they already decrease, they will stop the water molecule movement, and then they will tightly to like this. But for the solid, the hydrogen bond are stable, very stable, so that it will form the crystalline structure that have the whole one. There are banyak lubang dekat sini. They will not tightly to each other. No, they will not tightly to each other like the normal solid particle that you see. So hydrogen bonds are very stable, so it will form the crystalline structure like this. Due to there are many space at there, so that the density will not very high. The density will not very high. The density only very high, maximum, then not maximum at 4. This one is already become I, meaning that the temperature is decreased below the 0. Now, if you want to have a maximum density is 4. And then you go compared to liquid, the hydrogen bond will constantly break and reform. So yeah, this one is that you need to learn. So this structure make the ice can float on the water. It will not deep. The ice will not sink. You compare to other type of solid particle, any solid particle, if you throw into the water, then it will sink, right? It will sink. But ice as the solid particle float due to this property, due to the maximum density at 4 degrees Celsius, and due to the crystalline structure and stable hydrogen bond formed in the ice. So that ice will float on the water. And you compare to warm water, the warm water, of course, if you compare to warm water, the water molecule are not attracted to each other lah. But if you go to cold water, the water molecule will attracted to each other. Yeah. So most important thing I need to mention about this. And this one you will learn again in chemistry. Chemistry also you will learn about the formation of hydrogen bond. Also you need to know the reason. In chemistry they maybe come out. So yeah, you are very lucky lah. One knowledge you apply in 2 subjects. Okay, so this is just a summarization of the water molecule. So I hope this one is clear. So this is very easy topic, and I need to tell you until now no past year never come out yet. Never come out. Except objective maybe come out, but in structural question and essay question never come out yet. So you are the last batch. Maybe come out, maybe no, I don't know, maybe. So this is very easy. I hope that everyone can know it lah, and know it because this is, I can say it very easy lah. Okay, so next I already finished up the water part. So next I will enter the carbohydrate. Okay, this next chapter is about, next topic is about the carbohydrate. So I think carbohydrate everyone also know lah. What is carbohydrate? You every day you already eat one rice. So of course you are every day touch about carbohydrate. So if you still remember inside your SPM, you learn about you learn 3 components, right? Monosaccharide, disaccharide, polysaccharide. Similar to STPM, you also learn about these three. So totally same, no different. So we know that this is just a general structure of the carbohydrate. We know that carbohydrate is the organic molecule, right? It one type of the complex organic molecule, and also another name in general term is sugar. Sugar is the basic unit of the carbohydrate. But in exam you cannot write sugar, you need to say monosaccharide. So the main component atom of the carbohydrate is carbon, hydrogen, and also oxygen only these 3. Others, if got nitrogen, if got sulfur, all that, if that one is not say carbohydrate lah, not considered as carbohydrate. Okay, and then there are also the ratio of this atom is 1:1, meaning if they are 1 carbon, automatically they will have 2 hydrogen and also 1 oxygen. This ratio. And also the formula of this carbohydrate is CH2O-Y. Okay. And then the functional group of the carbohydrate, they are containing two functional group. Either is hydroxyl group or carbonyl group. Although you are not entering chemistry yet, but I assume that you already learned chemistry. Organic compound, right? You also learn alkane, alkene, carboxylic acid, carbonyl, ester, all that you also alcohol, hydroxyl group, all that you already learn in SPM, right? Chemistry. So you are not you don't have the alasan untuk cakap awak tak tahu lah apa itu what is the functional group. Okay. Now we go to this basic function. So we know that the basic function of the carbohydrate is to produce energy. We know that in your aerobic respiration, the first compound is glucose. You need to use glucose to produce energy. Right, so it provides energy. So this one, later you will learn more detail in chapter 5 about how the glucose is metabolized in our cell to produce energy. They have the metabolism. So this one I don't need to touch very much. Also, no, so if you go through the short-term energy storage, of course it's glucose. So of course glucose, but there are also the long-term energy storage. So normally it's excess glucose will be stored in form of the starch in chloroplast, also in form of the glycogen in liver and muscle. So for plant, they will store in the form of the starch, and then in animal they will store in the form of glycogen. Okay. So the role of the carbohydrate in plant also play the role in animal and plant structure. Not only play the role in produce energy, plant structure and animal structure are also very important. So for plant, there are also the glucose molecule also composed to form the cellulose. So we know that glucose, this one is beta glucose. So very important, you need to know there are two glucose molecule, alpha and also beta. So this is beta. What are the difference between alpha and beta? You see the first carbon, the hydroxyl group. The hydroxyl group of the first carbon. For alpha that you normally see in your SPM is at the lower one. Beta is at the upper. Ini memang kawan kena, you need to know, you cannot be confused. Alpha is at the lower, beta is at the upper. Very clear. Beta upper, alpha lower. Carbon number 1, not carbon number, not this, only this one. Okay, later I will show you more clearly. And also there another one is chitin, another form of carbohydrate, also composed of the shell component of arthropod. And then there are three main classes of carbohydrate that you see: monosaccharide, disaccharide, and also polysaccharide. So totally same. So we go through the monosaccharide first. So monosaccharide is the simplest unit of carbohydrate. So this is a monomer, the basic unit. The normally that you see is glucose. So what are the main property of the monosaccharide? One of the most famous property is taste sweet. It is taste sweet. Manis. They are basah manis and can able to soluble in water, right? So sugar can soluble in water, and they are colorless and can be crystalline. And they are also as reducing sugar. So this one, I think in SPA you also already know lah. It can act as the reducing agent and oxidized by the Benedict reaction. Benedict reaction, meaning that you see that your color, if there are the reaction, your solution will turn into the reddish brown color, right? Itulah, from colorless to go to the reddish brown color. That one that you already learned in SPM. So about the empirical formula of the carbohydrate, so as I mentioned before, CH2O(N) is the basic formula of the carbohydrate. So if you want to form a basic monomer of the carbohydrate, your carbon must at least have 3. Number 3. You need to have at least 3 carbon atom. Can baru boleh form one carbohydrate. The one monosaccharide, sorry. So there are normally basically there are 3 to 7. So number 3, 4, 5, 6, 7. So normally in general, as we know that glucose is considered as there are 6 carbon atom. There are 6 carbon atom in the glucose. So if we classify according to the number of carbon, they were like if carbon number they have the monosaccharide, they have the 3 carbon known as triose, and they have the 4 carbon tetrose, 5 carbon pentose, 6 carbon hexose, 7 carbon hexose. So normally in our daily life, most important is 6 carbon hexose. So glucose, galactose, fructose, all these are considered as hexose. And another one more important is carbon number 5. They have not carbon number 5. They have 5 carbon pentose sugar, ribose. This one is important in the nucleic acid, right? Nucleic acid, DNA and RNA. We know that the basic structure of nucleic acid is containing DNA, RNA, right? This one later you will learn it two in this chapter, they composed of ribose. Others are not very important. You don't need to know it. You just know about the several example. Then enough lah. Okay. So this is an example. We will compare to the structure formula lah. It like this. So classification according to the functional group. So there are already mentioned that the main functional group of this carbon is containing carbonyl group and also hydroxyl group. But hydroxyl group are not very important for the carbohydrate. But the most important is carbonyl. Carbonyl group is divided into two. Either is aldehyde or ketone. So as you guys know that aldehyde is where your carbonyl group is at the end of the carbon skeleton. Meaning that you see this example, your carbonyl group C double bond O is at the end of the carbon skeleton one. Dia dekat belakang punya. But if you compare to ketose sugar, your carbonyl group C double bond O is at the middle one. At the middle. So this is two main difference. So for the example of aldose like glycosidic, ribose, glucose, and galactose are example of aldose, meaning that it is containing aldehyde group. While another one is ketone. One of the main famous example is fructose. Fructose is the one of the example of ketose, ketone group. The carbonyl group is at the middle one. Okay. So yeah, this one is another. So you see the glucose, galactose, you focus on the six carbon hexose sugar. So glucose and galactose are aldehyde group. So aldehyde which we know as aldose sugar. Why? Because the carbonyl group is located at there. The difference between glucose and galactose is their location of the hydroxyl group. The hydroxyl group. So for glucose, the hydroxyl group is at the right hand side. You see, it is at the right hand side, but galactose is at the left hand side. The location of the hydroxyl group are different. But from there to fructose, the main difference is their carbonyl group. The carbonyl group is at the carbon number 2. At here, carbon number 2. This one, the carbon number 1 already formed carbonyl. This one is carbon number 2 at the middle part. Now at the middle part is but at the carbon number 2. So you need to know how to draw it. Memang awak kena tahu how to draw glucose, galactose, and fructose. This one you must know. And also ribose. Ribose also you need to know it. But glucose, galactose, fructose are very important. You need to know their differences and the location of the carbon atom. So for the glucose, galactose is carbon 1, 2, 3, carbon number 4. At carbon number 4. But this one, fructose, is carbon number 2. The carbonyl group, the ketone group is at the carbon number 2. Okay, so yeah, you need to know that. And also ribose you also need to know. Okay. So if you naming that like this so you name me according, this is a general name, just a general name. So if there are the number carbon 3 carbon and this one is aldose sugar, we know as aldose triose. And ketose, you go at ketone triose. So similar, aldotetro, aldopetro, aldohexose, like this these are the general basic And also arrangement of the monosaccharide. Okay, just now you see that it is in the linear form one. Later this one structure is in the linear form. The basic chemical structure that you draw in your SPM, right? SPM you draw like this, But actually, if all the sugar molecule, the monosaccharide, especially the carbon number 6, carbon more than 5, carbon number 5, number 6, number 7, all these they actually can form ring structure like this. That is why that you see like this. Why they can form ring structure when they dissolve into water. And only the ring structure, this form of the glucose monosaccharide can make disaccharide and polysaccharide. Meaning that this one, that triose sugar and also triose sugar, the glycosidic, dihydroxyacetone, all these all these and also tetrose sugar cannot form disaccharide and also polysaccharide one. They only can form the basic unit only. If you want to form disaccharide and polysaccharide, you only can form ring structure. So this one is that they will form the ring structure automatically when they dissolve in water. They will have the automatically reaction. So this is the reaction open. So, ini very important. You need to know how it does. How it dia macam mana tukar punya. Ini very important. This one is an example of alpha glucose. Alpha glucose. So the basic lah, alpha glucose. So the first important thing is you need to label the carbon first, the number of carbon. So this one actually you also already learn in STPM, right? How to number of carbon. Firstly, you is naming according to the functional group. The first one is the carbon. Carbon will be according to the functional group. So aldehyde group is at the bottom. So the carbon is carbon number 1, 2, 3, 4, 5, 6. Okay. So the most important thing is you see carbon number 1 and also carbon number 5. If you go to this, if you already form the ring intermediate ring structure, it will form like that. You will see that you will form the hexagon, right? You will form the 6 carbon, so you will form the hexagon structure. So you see that there are the carbon at here, So you see carbon number 1 like this, right? C double bond OH, and then go to C carbon number 2, HOH, carbon number 3, carbon number 4, carbon number 5. And then after that, carbon number 5 will bond to carbon number 6, CH2OH. But later, if you want to form the ring structure, how this carbon number 5 and carbon number 1 can interact with each other. Macam mana? So how do they interact? It will interact like that. So the C double bond or the carbonyl group and the hydroxyl group for carbon number 1 and carbon number 5 will interact to each other. Meaning that all the hydroxyl group, the oxygen atom will bond to the carbon number 1. Carbon number 1, and this hydrogen atom will go to this. To the oxygen at the carbonyl group at here. Meaning that C double bond or it will break up the double bond of the C double at the C double bond or to become the single bond. So if you form big single bond, you need to stable it. So how to stable? Just donate to hydrogen atom from here kepada sini, transfer to here, so that it will form another hydroxyl group at here. So another hydroxyl at here. So this oxygen atom, because the hydrogen atom is already hilang, tak stable lah, stable. So they need to form a stable bond. So it will form back the bond to the carbon atom. So this oxygen will form back the bond to the carbon atom. So it will form the ring structure like this. So this one you can see is like the hexagon structure, right? So hexose. So this is an example of alpha glucose. Another one, beta glucose, is that the hydroxyl group at the number 1 is at the upper one. But the similar structure, similar theory, just that the hydroxyl group will be at the upper one. You will see double bond or is located at the upper, not at the lower. Okay. So yeah, this one is the alpha glucose. So that why you can see this one is the ring structure form if you want to form disaccharide and also polysaccharide. So this one you need to know how to draw it, how to form it. Okay. Another one is fructose. How about fructose? Okay. So I need to tell you fructose is quite different. Why? Because fructose is ketone group at here, ketone. So meaning that the formation of the bond is quite different compared to aldehyde group. Glucose is aldehyde. This one is ketone. So meaning that E at the carbon number 2. So similarly, you need to draw all the structure, ring structure like this, ring forming structure according to the carbon number. But carbon number 2 will react with the carbon number 5. Dia memang akan react dengan carbon number 5. But the difference is glucose is carbon number 1 to number 5. But now is number 2 to number 5. So meaning that you will see there are carbon number 1 at here. Ca2OH, and then join with carbon number 2 and double bond O, and then carbon number 3, number 4, number 5, and another one form number 6. So they are OH2. So similarly, what you form this, similarly, same concept, you need to break down the carbon bond. See double bond O, you need to break down to become single bond. So once it becomes single bond, it unstable. So this hydrogen atom will transfer into this part, so becomes COH. So you will form like this, COH, and then this oxygen atom, due to the hydrogen bond is already lost, you want to form the stable formation, stable bonding. So this oxygen atom will bond to the carbon. So you will form like this. So that is why that fructose you will see like pentagon, whether they hexagon, due to this reason. So this one you also need to know how to draw it. So not same to the glucose. The main difference is here, carbon number 1 and carbon number 2. The main difference is here. Yeah. So yeah. So hopefully you guys understand and know how to draw it. Okay. Okay, next you go to function of monosaccharide. Okay, I think this one just a basic theory. So I think I don't need to talk much about this. You just read yourself lah. Okay, I hope that you can read yourself. So I don't want to talk much this lah. So okay, now we go to disaccharide. Okay, so disaccharide, we know that it is a carbohydrate, the monosaccharide that joined by two monomers through the condensation process by glycosidic bond. So we know that in SPM, you know glucose and glucose will form maltose, glucose and sucrose will form fructose, and glucose and galactose will form lactose. All that right? Or that you know. So similarly to the disaccharide, so you need to know about the properties. So it also soluble in water and also tertiary and also crystalline, and all of that is as the reducing sugar except sucrose. Why sucrose is not as the reducing sugar? Because sucrose does not containing any free ketone and aldehyde group. And we want to have a reducing sugar, you need to have a carbonyl group 1. If you does not have any carbonyl group inside your sugar the structure, meaning that it does not have the reducing sugar, no reaction as well. So this group, because the carbonyl group for the glucose and galactose are already formed involved in glycosidic bond. Later we will see how does this. So if you want to form disaccharide, I already say is include the condensation process, meaning that it will remove one molecule. The water molecule will be released out. And while the example, this one is maltose. So when there are the alpha glucose and alpha glucose combined together will form maltose, right? So the most important thing is I need to emphasize is this glycosidic bond. The bond between the glucose and glucose, between the monosaccharide are known as glycosidic bond. And you need to write alpha 1,4 glycosidic bond. Alpha is due to the type of the glucose, right? Is alpha glucose, and 1,4 is the location of the carbon that form bond. 1 is the carbon number 1 at one glucose, and 4 is another glucose that form carbon, that form bond. So at carbon number 1 of one glucose and carbon number 4 of another glucose play responsibility to form the glycosidic bond. So that is why that alpha 1,4 glycosidic bond. So this one you also need need to know how to draw this equation, not how to draw the structure, full structure. Just not say glucose plus glucose, condensation, maltose plus water. No, you need to draw the original structure like this in exam. If exam required to draw, explain the formation of maltose, you need to draw the chemical equation like this. You cannot write the chemical equation just like glucose plus glucose, condensation, hydrolysis, and maltose. If you draw like that, no marks lah. Now mark you need to draw like that, because the key point is that you need to show examiner need to understand, want you to understand know whether you are understanding about the formation of glycosidic bond or not. The most important thing is at here. Not only maltose, we know that lactose and also sucrose. So I will focus about the sucrose. So we know that sucrose is the formation of the between glucose and also fructose. And you know that glucose is aldehyde group. They are the carbon number aldehyde group at here. And then fructose have the have the ketone group at here. So when they are the undergo condensation, they will produce like this. So they will form alpha 1,2 glycosidic bond. And at here, there are no any free aldehyde and ketone group already. Why? Carbon number 1 and carbon number 2. You see this, you will see maltose, carbon number 1, carbon number 1, that aldehyde group, one of the glucose carbon number 1 that containing aldehyde group already play the responsibility for formation, but another one no. Tak lah. The aldehyde group still remain at here. So meaning that this one will act as the reducing sugars. They still can reduce the reducing sugar, meaning that it also can act as the reducing agent to reduce substance. If you test about the Benedict test reagent, they still can have a positive reaction, meaning that the big V formation will still form in the solution. But if you go to the sucrose molecule, due to the carbon number 1 of the glucose and carbon number 2 of the fructose, both of them play the role in the aldehyde group and also ketone group are already formed in the glycosidic bond. So for sucrose, they are already do not have any free aldehyde and also ketone group. So that is why that sucrose molecule cannot play the role in reducing sugar. Okay. Another one is lactose. When you formation of the bond of the galactose and also glucose, it will form the lactose lah. So concept similar to get glucose and glucose lah. Okay. Okay, so I hope everyone can hear. And another one is oligosaccharide. Although this one is not in the syllabus, later I need to just want to let you know that before polysaccharide is very highly complex. But there are also the oligosaccharide. Oligosaccharide is that the carbohydrate is made up of small number of monosaccharide. Compared to disaccharide is 2. But oligosaccharide is normally is 10 to 10 only. 2 to 10 like that. Smaller than polysaccharide lah. But this one I don't need to explain much because it is not in syllabus. I just want to tell you soluble in water, test street, and classify based on the number lah. And this one is the example of the oligosaccharide. Okay, and then the example lah. You don't need to know it. Not important. Ini example you don't need to know it. Okay. Now I'll go to the polysaccharide. So this one is very important. Very important lah. Very important. Ini important lah. The oligosaccharide no important. You don't need to focus it. Polysaccharide is very important. Very important. It is a very complex and very large in size of carbohydrate. So main property of polysaccharide is it is insoluble in water. Compared to mono and monosaccharide and disaccharide, both of them are soluble in water, and both of them can be crystalline. This one cannot be crystalline. So polysaccharide, one of the example is rice. When you eat rice, rice is carbohydrate, right? The main every day you eat. Do you taste rice is sweet? No, it is not sweet. Not sweet. Why? It is not taste sweet. And another one is it does not non-reducing sugar. It will not have the free aldehyde group and free ketone group as well, because it is very complex structure. Later we will see it. Okay, so polysaccharide be classified into two either we classify into homopolysaccharide, and also heteropolysaccharide. So in our syllabus, we normally focus on homopolysaccharide only. Heteropolysaccharide is not our syllabus. Homopolysaccharide. And polysaccharide also considered as either is branched or unbranched. Very important. Unbranched or branched. So I just want to tell you now, in our syllabus, we only learn three examples of polysaccharide: starch, glycogen, and also cellulose. These three only. Others you will not learn. You don't need to result. I will result in our syllabus. We only learn three: starch, glycogen, and also cellulose. Both of them, three of them are considered as homopolysaccharide. So what is homopolysaccharide? Meaning they are only made up of one type of monomer, one type of glucose, one type of monosaccharide. All of them made up of glucose. So more diabetic glucose. All of them are made out of glucose. Okay. And then the bulk of the function of polysaccharide. So normally one of the function is energy storage. So in animal is a glycogen stored in form the glycogen, and in plant is formed in the starch. So it makes the cell more hydrophobic due to the formation of hydrogen bond between the monomer. Later we will see it, and lower the sugar, this lower the sugar concentration. So that if you compare to glucose is the solid form, only one form. But we will compare it into the one form. Compare it, so it will lower the sugar concentration. And also the cellular communication can bind to other biomolecule like lipoprotein. Glycolipid, glycoprotein is also one of the example of polysaccharide. And also cellulose support cell component of a plant and also fungi and also protect the cell from damaging. Not only plant and fungi, also bacteria. Bacterial cell also containing polysaccharide, to protect . Okay. So example of the polysaccharide. Okay, as I mentioned before, you need to learn about starch and also glycogen and also cellulose. But before we go to it, we need to learn about the formation of the polysaccharide first. Meaning that you know that polysaccharide is the formation of the combination of 100 of the glucose molecule into very long chain. And as I mentioned before, you have branched and also unbranched. So if you want to form unbranched chain, unbranched polysaccharide, how to eat? We know that if you want to form glucose and glucose form maltose, is using alpha 1,4 glycosidic bond, right? Similar concept. This one, if they are very long, we will not say that maltose anymore. We say as amylose. Meaning that amylose is an unbranched polysaccharide consists of alpha glucose that link together by alpha 1,4 glycosidic bond. So when there are many maltose, you can imagine when there are many maltose more combined to each other, they will form amylose. And this one is unbranched, tak branch, panjang je. Dia dekat satu line je. Satu line je. Macam ni lah, satu line je. Dia tak keluar sini satu, tak ada. Satu line je. So this one will cause the helix structure to be straightened. Another one I need to focus is branch structure. Branch structure. We call as amylopectin. Okay. Amylopectin is that it form the branch structure. So this one, if you see ini satu line ni, this bottom part is amylose, and this one is also contained also as amylose. Amylopectin is that whole substance. This whole, when there are the branch at here, we call as amylopectin. If we just look a single line, we know as amylose. Amylopectin is whole structure. A whole structure that containing a branch one, we call it as amylopectin. So it is a branch polysaccharide. So this branch polysaccharide, how it's formed? The hydrogen bond is formed. The bond formed is alpha 1,6 glycosidic bond. not rather than alpha 1,4, not alpha 1,4, 1,6 already. Meaning that there are the carbon number 1 at here and carbon number 6 at here, form the bond. Rather than carbon number 1 and carbon number 4. Carbon number 1 and carbon number 4 only form the amylose. Carbon number 1 and carbon number 6 will form amylopectin, which branch. So this gave a more open molecular structure and prevent helix formation. And then also amylopectin can be hydrolyzed more easily and slightly more water soluble compared to the amylose. Okay, so this one is more single lah. Amylose like this. Malonyl will form helix structure one. Akan berbentuk helix structure. And then this one is amylopectin. Like this. More complicated lah. You will see that this one amylo. In another word, amylopectin is a combination of the amylose. Banyak amylose at here. One amylose at here and another amylose at here, and between amylose they will have the bond we call as alpha 1,6 glycosidic bond at here. Okay, so yeah, this one is starch. So compare the difference between starch and also the glycogen is their branch structure. Starch is not complete highly branch structure. The main structure of the starch is amylose rather than amylopectin. Amylopectin, yeah, present but very low amount. But if you see glycogen, yeah, glycogen is very complicated. It is highly branch structure. There are many branch at there. So you see this diagram, wow, many branch. But you compare to starch, it is more arranged, into the line one. There are also the branch, but very little. So starch is the energy storage for plant in chloroplast. So yeah, this is the explanation. Okay. Yeah, in glycogen, it is energy storage in animal liver. The main difference is it highly branched. There are many amylopectin structure. Amylopectin structure is very, very high. So yeah, so it is a highly branched polymer structure of alpha glucose residue. It also made up of alpha glucose. Glycogen and starch, both of them are made up of alpha glucose. Both of them are alpha glucose. Okay. So that is why that glycogen is also known as animal starch, because of its similarity to amylopectin, except that is made up of shorter chain and highly branched. So comparison between starch does not have many amylopectin. The branch is less, but it is very long. The amylose length is very long. The chain is very long. Compared to glycogen, it is highly branched, but the carbon chain is very short. Okay. So that is why that glycogen is less dense and more soluble than water. So it is a more suitable storage for animal. So glycogen clump together to form small granules and are most often stored in liver and muscle cells, and also break down easily into glucose by enzyme glycogen phosphorylase. This one you also learn in semester 2. Ini mechanism you will learn in semester 2. Okay. The last one is about cellulose. Okay, cellulose is very spatial skin. Why? Because it is used beta glucose rather than alpha. We will use beta. So I already mentioned alpha glucose and beta glucose. The difference is the carbon number 1. Carbon number 1 at here. Carbon number 1 at here. So carbon number 1. So if you alert that the carbon number 1 for alpha, OH is at the lower part, but for glucose, OH is the upper part, at the upper one. Is at the upper part. So why like that? Why you want to use at the main component is beta glucose, and the formation is bond is beta 1,4 glycosidic bond. And if you refer to this structure, you can see macam pelik-pelik punya. You will see the ada pelik punya structure. Maybe you will not see later. You see this, you will see the difference. The main difference is at here. You will see carbon number 1 and carbon number 4 at here. Number 1 and carbon number 4. This one is beta glucose. Glucose. You will see that you form number 1, 2, 3, 4, 5, 6. Carbon number 1, and then it form another bond at carbon number 4. Carbon number 4, another beta glucose. But carbon number 1 is not at carbon number 1 is calculated at here. 1, 2, 3, 4, 5, 6. Reversible. You see the difference. If you refer to the alpha glucose, all the carbon is calculated as 1, 2, 3, 4, 5, 6. It's calculated like this. It's similar to here. 1, 2, 3, 4, 5, 6 is for alpha glucose. But for beta glucose, the carbon number is calculated reversible. Meaning that successive beta glucose are oriented 180 degrees Celsius to each other one. Meaning the carbon number is calculated number 1 starting at here too, but not at here. This one is not carbon number 2. You see the oxygen atom is here, meaning that it's calculated 1, 2, 3, 4, 5 and 6. Totally terbalik. Memang terbalik. So that is why oriented 180 degrees Celsius memang terbalik. So that is why there is still form 1,4-glycosidic bond rather than 1,6. This is one of the reasons because they were arranged, oriented reversibly to each other. So that is why there is still form 1,4-glycosidic bond. Beta and the main, you need to mention beta. And you don't know, you will only write 1,4-glycosidic bond, no marks. You need to mention beta. Similar to amylose and amylopectin, you need to mention alpha. Alpha 1,4 glycosidic bond for amylose, alpha 1,6 glycosidic bond for amylopectin. Okay. So cellulose chain run parallel to one another. This one is one of the structure. Cellulose does not obey any amylose and does not obey amylopectin too. They obey the unique one is that they form the beta 1,4-glycosidic bond. The glucose molecule, the beta glucose is oriented to each other, reversibly to each other, and they arrange in parallel form like this. They were not interconnected to each other one. They were interconnected to each other through hydrogen bond only. No glycosidic bond at each other one. At each parallel line each line. No. They only interconnected to each other through 1,4-glycosidic bond only. 1,4-glycosidic bond only. They will not have a branch structure. They just only one line. And between the line, they will have the hydrogen bond hold them to each other. So this is totally unbranched structure. Cellulose is unbranched. So these chain together to form microfiber, which are arranged in the larger bundle to form a microfiber, and this will give plant cell high tensile strength and rigidity, which prevent the cell from rupturing when water enter by osmosis. So cellulose is the main component of the plant cell wall. Later we will explain more detail about the structure of the cell wall in chapter 2, but this one, cellulose, is one of the important things due to this structure. One of the important key characteristics provide the plant more strong and more flexible and more rigid. And that is why that in hypermorphic solution, the plant cell will not burst compared to animal cell. And human cannot digest cellulose. That one of the reason is we does not have cellulase. Only the some of the animal have cellulase, but human cannot secrete cellulase one. Our gene, our body cannot produce cellulase. So that is why that when you eat the vegetable, especially when you eat the more harder vegetable, you will feel that very difficult to break down it. Why? Because we does not have cellulase. It will directly totally come out from the body without any digestion process. So this is the structure of cellulose and then this one is the spatial chitin. This one is example of the heteropolysaccharide. One of the example of heteropolysaccharide. Okay, but you no need to learn it or not in your syllabus. Okay, so we proceed to lipid. Okay, so you need to know what is lipid. So lipid, if we according to the atom, the organic chemistry, so normally lipid containing carbon atom, hydrogen atom, oxygen atom, nitrogen, and also phosphorus atom. The difference between water and also carbohydrate, lipid does not have the fixed ratio one. There are no fixed ratio for the lipid. So there are many, you can say there are many manner to control the lipid. So lipid containing carbon, hydrogen, oxygen, nitrogen, and phosphorus. So normally if you remember about the ester, right, you still remember the SPM that you learn about the hydrocarbon chain, that chapter, you learn about ester, right? So ester actually is one type of lipid too. One type of lipid. So normally lipid we are classified into either this it is the saturated fat or unsaturated fat, and then the common functional group are ester. So ester. So ester actually is if we change to common name, lipid lah. Because lipid containing the ester functional group. So we still remember what is ester functional group. It is a combination of carboxylic group and also hydroxyl group, right? You still remember hydroxyl group and also carboxyl group to form the ester. Okay. So normally lipid property has these all properties, including they are maybe liquid or maybe solid form at room temperature. So you need to see what type of lipid, what type of component. So like saturated fatty acid and also unsaturated fatty acid, right? This one later we will see it. And also pure fat and oil are normally colorless, odorless, and tasteless. If we consider pure, you cannot see that that you see in your kitchen food oil, right? That you use the oil during cooking, or you using the fried chicken, fried rice, all that. During the cooking, your oil is already been processed, already got additional chemical or additional substance, additional organic nutrient, all like that. So it can change to yellow color that you see in the kitchen. But actually, if you want to see pure fat, totally pure, 100% pure, there are actually colorless one. They are colorless, similar like a water one, and also odorless and also tasteless. Actually, lipid fat does not have taste one. Yeah, right. Right, if you only drink test the oil, the cooking oil, any cooking oil, you will find that there actually no taste. No taste one. So that is why you cannot drink oil. If you drink oil, actually no such thing because it is tasteless. And also there are one of the property is there are energy rich organic molecule. It is highly energy rich compared to other type of biological molecule, especially carbohydrate. Carbohydrate. And also another thing is it insoluble in water. Compared to carbohydrate, if you see monosaccharide and disaccharide, normally can dissolve in water one. They soluble in water, except polysaccharide. Polysaccharide cannot soluble in water. But any lipid is insoluble in water. But they are soluble in organic solvent. So like alcohol, acetone, benzene, all these, especially alcohol. If you pour the oil into the alcohol, oil will disappear. You will not see the oil in the alcohol liquid one. Compared to water, if you pour the oil into the water, you will see 2 layer, right? Oil will be at the above layer. Why? Because oil cannot soluble in water. And another thing is there are no ion charge. Compared to no ion charge. Okay, so and then this one is the general function of the lipid. So normally the main function is energy storage. You already mentioned it is the rich organic molecule, right? Organic molecule, so it will play major role in the energy storage. And another function is to regulate hormone, because some part of the hormone actually is considered as lipid. So for example, like estrogen, testosterone, and also cortisol. These all hormones are lipid. These all hormones are lipid. They are actually considered as lipid, under category of lipid. So they are regulate hormones. So meaning that they are part of the production to produce this kind of hormone. And also transmit nerve impulse. Lipid can generate myelin sheath, which can speed up the rate of impulse transmit. This one later you will see in chapter 2 about the nervous tissue, but I think this one you also learn in SPM lah, but I don't know you still remember or not. The nervous tissue, you know, like animal tissue we contain epithelium tissue, nervous tissue, connective tissue, all that, right? One of the tissue is nerve tissue. Myelin sheath is one of the component that on the neuron that can speed up the rate of impulse transmission. Another one is cushion vital organ. It provides a cushion to help to protect our organ. So normally this is just the it's just like a layer. We can say that it is simply like a layer to protect our organ, right? Our internal organ, especially in our stomach, because you know that in our stomach area we do not have bone. We only containing skin and also adipose tissue only, which is fat only. This fat actually protect our internal organ, especially stomach, your small intestine, large intestine, pancreas, all that. Because at that part no bone. There are no bone to protect us. Compared to lung, if you see the lung we containing rib cage, right? Rib cage can protect our lung and also our heart. But in our stomach, with the organ within the stomach, there are no bone to protect. So fat tissue can provide the cushion to protect our organ. So it is good, but if too much, then it's not good lah. Okay, and maintenance of temperature, of course, if you angular temperature, of course, because fat act as insulator. This one is very easy. If you go to aircon room or you go to the very cold country, you can see that the people which is very fat, they are not easily to get cold compared to the people which is very kurus lah. Which is very thin or fleet. So that people will be easily to get cold. Mereka rasa sangat sejuk punya. Tapi berbanding kepada orang yang gemuk sikit, mereka tak akan rasa sejuk lah, walaupun dalam temperature yang sama. Okay. And then this one is transport fat soluble protein. Okay. And then intra and intercellular signaling event. So normally this one can be act as the receptor lah. And final one is serve as structural component of biological membrane. One of the example is phospholipid lah. Right, phospholipid. You know that you although not entering chapter 3, but in SPM you know that you got one chapter is learning about the plasma membrane, right? You know that the major component of the plasma membrane is phospholipid. So why it is called a the terms already mentioned, right? Phospholipid already got lipid, so the major component is lipid. Later we will see the structure of the phospholipid too. Okay, and then now we consider to the structure of lipid. Okay, if in lipid there are two general structure, two main structure. Either it is open chain structure that you see in triglyceride, like fat fat, fatty acid, all this is the open chain structure, hydrocarbon chain one. But there are also fused ring structure like this. This one is also considered fat. They are also not considered fat, also considered as lipid, but it is in fused ring structure. You can see this in the cycloalkane, or benzene structure. Okay, benzene structure. So this one is we call as fused ring compound. So normally the open chain one is normally insoluble in water and soluble in organic solvent, as mentioned before. And another one is amphiphilic hydrocarbon, and also useful for long-term energy. So yeah, so we will see it one by one. So this one is the most famous. Everyone should know already. Triose. This one, another name is triglyceride, and common name is fat triglyceride. But similar, triacylglycerol or triglyceride are same. Okay, now we go to fatty acid first. So we go to open chain first. We go to the open chain fatty acid. So I think fatty acid, you know that if you want to form the triglyceride, you need 3 fatty acid and also 1 glycerol. So we see the glycerol first. So normally glycerol, you can see that it is the 3 carbon alcohol compound that containing 3 hydroxyl group. So you can see that there are 3 carbon. So normally if you still remember, if 3 carbon, what is that? Methane, ethane, right? Methane, ethane, then propane, right? Propane, and then butane, and then pentane, all that, right? That you already learned. So this one actually, if there are no hydroxyl group, it is pentane. But now there are 3 hydroxyl groups, so this is one of the alcohol lah. You can consider as alcohol. So normally glycerol is the 3 carbon alcohol that containing 3 hydroxyl groups. So like that lah. And then another one is fatty acid. Okay, so fatty acid is long hydrocarbon chain that containing carboxyl group. So you can see this general structure. C carboxyl group is C double bond OOH, right? COOH, that is that you write in your STPM, right? COOH, but actually is in the C double bond O and hydroxyl group. So normally R will be the hydrocarbon chain. This is a very long hydrocarbon chain. So in fatty acid, there are no fixed hydrocarbon chain, but different type of fatty acid will have different type of carbon chain. So normally the hydrocarbon chain, the number of carbon is normally is 9 or even up to 10 also have. Yeah, also more than 10 carbon in one hydrocarbon chain. So although COOH, the hydroxyl group, is polar, but due to the hydrocarbon chain is very long, and this is a non-polar molecule. So this structure give that fat cannot soluble in water. Because due to this long hydrocarbon chain, they already cancel the effect of the polar group. So that is why that fat cannot soluble in water, although they are containing the polar functional group. However, due to the non-polar interaction is too high, already cover up the polar interaction, so that the fatty acid, the fat cannot soluble in water. And then this one is function of fatty acid. Okay. So one of the function is the building block of the lipid lah. Lipid, modification of protein, regulation of membrane unity, serve as hormone and also intracellular message, and final one is energy storage lah. And then as you know that there are 2 types of fatty acid: saturated fatty acid and also unsaturated fatty acid. So the key difference is the double bond. Saturated fatty acid is all of the carbon inside the hydrocarbon chain are single bond. there are no double bond or triple bond in each other one. So you see this structure totally single bond. There are no double bond except the carboxyl group. But we consider as a hydrocarbon chain, there are no double bond or triple bond. But unsaturated fatty acid is the acid that carbon chain, the hydrocarbon chain containing at least one double bond, at least one. Not necessarily only one, but they are at least one. Maybe one, maybe two, maybe three, maybe four, like that. They should at least one double bond group. So you can see that in this structure, unsaturated fatty acid, they containing double bond at here. And then in unsaturated fatty acid, as I mentioned before, they are either containing one double bond or they are containing more than one double bond. So normally, if there are only one double bond in the fatty acid, we call as monounsaturated lah. Another one is when there are more than one double bond, maybe two, maybe three, maybe four, more than two lah, or they are considered as polyunsaturated fatty acid. And also a ratio of unsaturated fatty acid is different compared to the saturated fatty acid. Saturated fatty acid is totally straight chain only. They are totally straight chain. So saturated because they are single bond. All the carbon is filled up with the hydrogen atom. So they will produce the only straight chain. Garbage to lose the. But in unsaturated fatty acid, due to the presence of double bond, they can be either bend shape or straight chain. Bend chain or straight chain. Based on the structural form lah. Either this is the cis form or trans form. So if the hydrogen the structure is cis form meaning that the hydrogen atom is located to side to each other at the same side. You see this double bond, right? The hydrogen carbon are totally at the same side, either at upper part or bottom part. One of them lah. But if the hydrogen carbon are arranged to each other at the same side, so this one is cis form, they will produce the band shape. This one is called kin structure. And then this one is very important in terms of the plasma membrane. Later you will see it in plasma membrane. This structure is very one of the function because it can prevent the unsaturated fat packaging together closely to solidify at room temperature. So that is one of the reasons why unsaturated fatty acid is liquid form in room temperature. One of the reason. But if the unsaturated fatty acid is in the trans form, meaning that the hydrogen atom are different side at each other, so it will like the saturated fatty acid it will be straight chain. Fatty acid also classified into either essential or non-essential lah. So normally essential fatty acid will be fatty acid that must be obtained from diet lah. We cannot synthesize it. But non-essential can be synthesized in our body. Our body can synthesize this one, but essential cannot. You totally need to consume out, similar to the protein amino acid. Amino acid also have essential and also non-essential. But essential is you must take from diet. So what normally is example is the plant oil like omega-3, omega-6, all that, or plant oil, fish oil, all that normally is you need to obtain from the outside. Your plant, our body cell cannot produce this kind of fatty acid. Okay, so this is a general difference of the fatty acid between unsaturated and also unsaturated. So while the major difference is number type of bond saturated, no double bond, only have single bond. But unsaturated, they are containing at least one double bond. Due to this, so that your number of hydrogen atom for saturated will have higher number lah. It is maximum number lah. But unsaturated, because of double bond, so that it containing fewer hydrogen atom. And the melting point for saturated is high, unsaturated is low lah. And another key difference, I think everyone know, saturated is solid form, and unsaturated is liquid form. in liquid form. So another sources normally saturated is animal fat lah. Any kind of animal fat, including human, normally is saturated. So, but another one is plant oil or fish oil. They are unsaturated. So normally olive oil lah. Another one effect in humans. So normally we know that saturated fatty acid is bad fatty acid, right? It is the bad fatty acid. So normally if you consume high amount of saturated fatty acid, you will get the cardiovascular disease lah. But unsaturated fatty acid is good for health, but not necessarily say that you eat many unsaturated fatty acid is good. No, but if you compare to saturated in the same amount, unsaturated is more healthy lah. That is why that many people nowadays that focus on health, they use plant oil rather than animal oil lah. Especially in house cooking. But in outside, normally they are still using the plant. They are still using the animal oil. But some of the restaurant, especially at the very high level one, normally they will already change to plant oil. And also effect of the hydrogenation. So normally saturated fatty acid is no effect lah. Unsaturated have effect. They can be converted into saturated fatty acid one. And in terms of similarity, another one is insoluble in water lah. And also hydrogen and oxygen are present in the chain, involve the formation of triglyceride. Both of them can form triglyceride one, and made up of carboxylic acid that containing carboxyl group and act as an energy provider to store energy. Now we go to triglyceride. So as you already know what is glycerol, what is fatty acid, now we need to know how the fat is formed. So fat is formed, when the fat is formed, is called as triglyceride. Another one is triacylglycerol lah, but you can write triglyceride is okay. Okay, so normally triglyceride is formed between the condensation reaction between glycerol and also fatty acid. So one glycerol react with 3 fatty acid, they will form triacylglycerol, which is triglyceride lah, one fat molecule. So if you compare to the high carbohydrate, especially carbohydrate, fat, you can see that fat is highly rich molecule. Because carbohydrate, although it maybe polysaccharide is complex, but this one fat is just a basic unit only. They already can form the complex structure like that. So this one is the condensation reaction, meaning that water molecule is released out from there through the hydroxyl group of the glycerol and also carboxyl group of the fatty acid lah. So they will form the ester bond at here. So that is why that lipid is also one of the example of ester. Because this is another example of esterification that you already learned in SPM lah. So why esterification process lah? So yeah, okay. And then also triglyceride also can lipid fat can also undergo hydrolysis. You can break down the fat through to release the glycerol and also fatty acid through the hydrolysis lah. So you need the water lah. But actually normally you need to catalyst lah. Normally you need the catalyst. One of the main catalyst is lipase lah, lipase enzyme. Okay, so normally this one is the function. So normally similar to the lipid, normally energy storage, thermal insulation, soap making, and also organ protection. Okay, so saturated fat and unsaturated fat normally is similar to the difference between saturated fatty acid and also unsaturated fatty acid. Okay, now we go to the phospholipid. Phospholipid. So another terms name you also can consider as phosphoalkylglycerol lah. So this is the type of lipid that has the similar structure to glyceride, but the difference is they are in terms of instead of 3 fatty acid chain, they are containing 2 fatty acid chain only, and another one is replaced to phosphate group. Yeah, so that is why that you see the phospholipid is like that. There are only two tail at there. These two tail are considered as represented as the fatty acid chain. So two tail meaning there are two fatty acid chain. But there are another one is not fatty acid, is we consider as phosphate group. So if you see this diagram, this diagram, so another terms of the phospholipid is lecithin. This one is that lecithin. So this is the basic unit if you want to form phospholipid. It's like that. Lecithin, because they are has a choline group attached to the phosphate group at here. One of the example is due to the phosphate group. There are the phosphate group at here. So this structure is called as lecithin. They are soluble, water soluble, emulsify, and membrane constituent. So this one is the difference is they are partially soluble in water one. Compared to the fatty acid, the fat triglyceride is totally cannot dissolve in water. They cannot soluble in water one. But this one is partially. Why? Because of this one is polar group. And these two, of course, non-polar. So this one is the difference. So you can see that phospholipid, they containing one phosphate group at here. Phosphate group at here. So you will see the triglyceride, there are no phosphate group. 3 of them are fatty acid. So phospholipid only containing 2 fatty acid. So that is why that if you see the phospholipid, they will have hydrophilic head and also hydrophobic tail. Hydrophilic head meaning it is soluble in water, right? This one is face to the water one. And another one is hydrophobic tail. This one is repel water. Repel water one. So that is why that you see the phospholipid arrangement is like that. At the outside of the surface is containing water molecule. Fatty acid does not like it, so the hydrophilic head will exposed to that lah. Similar to the lower part, why is reversible? The hydrophilic head is at the face into the cell, because inside the cell they containing water. So hydrophobic tail, the fatty acid is not like water, they repel water, so the hydrophilic head will expose to that water lah. So normally, if you see the behavior of phospholipid in water, as I mentioned before, they are philic and also phobic. So normally, phospholipid is, you can say that it is partially soluble in water. They are not totally soluble, but they are partially soluble. The hydrogen ion dissociate from phosphate group. This causes the phosphate to be the negatively charged. There are the reaction. If you still, there reaction occur when you dissolve in water lah. So that is why that when you arrange the phospholipid in water, they arrange at like this in form of phospholipid bilayer. That is why that there are 2 layer, bilayer, 2 layer at here. Why not just only 1 layer? Because if there are only 1 layer, inside the cell also containing water. I already mentioned fatty acid is not like water. They are repelling water one. They repel water. So they cannot expose to water, so that they will arrange in terms of phospholipid bilayer. Another one is they are in terms of the micelle and also membrane bulk vesicle like that. Okay. So you see in plasma membrane, for hydrophilic head will face to the cytoplasm and also extracellular fluid. While your hydrophobic tail will hide inside the layer of the hydrophobic center. Okay, now we already finished up the open chain lipid. These are all the open chain lipid. So there are 2 open chain lipid that you need to learn: fat, triglyceride, and also phospholipid. Phospholipid, another term is lecithin. Okay, now we go to the fused ring structure. This is the fused ring structure. We call as steroid. Steroid. I think everyone know what it. Maybe heard about it. What is steroid. So normally if you go to hospital, yeah, some of the disease there, doctor will tell you, right, or when your immune system is weak or something disease, they will inject steroid to you. So what is steroid? So normally steroid is the fixed ring structure of the lipid. So there are many examples of the steroid. So these are the one of the example, 5 example, common example that we normally see in our life. So all of us have this hormone except the sex hormone. So all the sex hormone are under steroid. So you can see progesterone, estrogen, also testosterone. So progesterone and estrogen, both of them are female sexual hormone, right? That play the major role in menstruation, and testosterone is a key hormone for male. And also there are aldosterone. This one is play the role in homeostasis. And another one, the most famous one, cholesterol. Right. So you can see in our syllabus, if we refer to MPM syllabus, the main we focus is cholesterol. The other you don't need to know the structure, but except cholesterol, you need to remember the structure. You must remember. You need to remember or how many chain. You can see there are the 1, 2, 3 hexagon structure and also 1 pentagon structure. And the number 2 hexagon structure, they are containing the double bond at here. And you need also know that there are the hydroxy group, there are the methyl group, methyl chain, and there are the hydrocarbon chain at here. You need to memorize it. Why? One of the year in essay, MPM asks you to draw the structure of cholesterol. With the aid of diagram, the question is like that. With the aid of diagram diagram, explain the structure of cholesterol. Yeah, they need you to draw a cholesterol. So you must memorize it lah. Others you don't need to do. Because in our syllabus they only mention cholesterol. The other I just let you know only. Aldosterone, progesterone, estrogen, and also testosterone are the example of the steroid. But cholesterol is you must remember. Okay, so this is just a repeat. Yeah, cholesterol. So the cholesterol structure is, there are 4 hydrocarbon chain. 1, 2, 3, 4. There are 4 hydrocarbon fused chain, fused ring. Is with the hydrophobic tail at one end of the hydrophilic region, and also OH group at another end. You need to know that this one is hydrophilic region and hydrophilic region, and this one is hydrophobic region. You need to know hydrophobic and hydrophilic. You need to label. You need to label too. To got marks. If you not label, you just draw only, no marks, or maybe 1 marks only lah. You cannot get full marks. And the importance of cholesterol in health normally is this. So I think this one everyone should know lah. Why cholesterol is very important. So normally one of the function is the formation of cell membrane to maintain our healthy skin. Also, require production of male and female sexual hormone. Why? As you know that cholesterol is steroid, and you see the other structure of steroid almost similar to cholesterol. You can see almost similar. The hydrocarbon chain is totally same. There are 2, 3 hexagon and also 1 pentagon. Totally same. The difference is that the functional group only. Maybe the functional group, you see the testosterone, the function there. This part is not hydrophobic tail. This one is hydroxy group, and this one is carbonyl group, and this one you can see. So meaning that, an example in simply way, cholesterol can be undergoes the certain chemical reaction, certain enzymatic reaction to convert into these all things. So that is why of the function is say they can produce male and sexual female hormone. Also formation of testosterone. Of course. I already mentioned at here. You can see structure totally same. The mother carbon is totally same. The difference is just a side chain only. You can see the difference of the cholesterol just at here. This one is the carbonyl group. This one is hydroxy group, right? hydroxyl group, carbonyl group, they can change, and you can see the carbon bond. One of the carbon bond is at here, and one of the carbon bond is at here. They can change one. And this one is same, and you can see the end of the here is also changed. But I mentioned, undergoes certain enzymatic reaction, cholesterol can change into aldosterone, similar to progesterone, ketone, and also estrogen. Yeah. Also formation of vitamin D in skin, and also formation of bile salt, and maintain stability and permeability of plasma membrane, and metabolize lipid, and also dissolve lipid soluble vitamin. So all this lah about the cholesterol in health. So I think that's all about the lipid. Okay, now we proceed to the protein. Okay, as I mentioned, protein is one of the famous topic. Mostly MPM come out, especially in whatever lah, essay question, structure question. The most topic lah in chapter 1. Normally in chapter 1, if MPM one come out topic in chapter 1, normally is protein. Compared to other type of biological molecule, protein is the most famous topic to be coming out rapidly in exam question, real exam question. So this topic is quite long. Yeah, it's quite long topic, and most important I can say this chapter. But not say that other chapter is not important. Yeah, also important but this chapter is one of the topic that MPM always coming out in essay. Okay, so protein. Okay, so I think I don't need to talk much. Protein, as you guys know that the basic function, the basic unit of protein is amino acid, right? Amino acid. So this is the structure of the amino acid. You can see that basic amino acid structure is they containing the carbon at the center. Yeah, at the center, carbon at the center, and they're containing two groups. They are containing amino group, and also carboxyl group. So that is why that is called as amino acid. Amino, this term is coming from amino group. Acid is come from the carboxylic acid. Right? Because right. Okay. So amino group is NH3 lah. Amino, right? Amino normally is NH2, NH3 sorry, and carboxyl is COOH. OH. So there are one amino group at here, and H2 bonded with at the center, and also carboxyl group bonded at the center, and there are the R group, which is a side chain. And we know that there are total in real world, whatever organism, there are only 20 amino acid in the world. Only 20. There are no more than 20. Only 20. Compared to carbohydrate, compared to lipid, there are more than 20 lah, maybe more than 20. But amino acid, there are only 20 amino acid only. 20 only. Yeah, only 20 only. So so meaning that there are 20 different R. Yeah, 20 different R. Different 20 R lah, we can say that. Okay, so this one basic structure, you must know it. You need to know it, how to draw it. Like that lah. COOH and H2, and there are the H at the bottom, and also R group at here. So what is protein? So I mentioned monomer is amino acid, and there are 20 different type of amino acid and can be combined to form a protein. And so protein, why protein is very complex? Because it have a larger range of the function. There are many functions. So what can you imagine what function? What function? If you still remember what function that you learn from SPM. One of the key component is enzyme. You know that enzyme is protein. Enzyme is protein. And then beside enzyme, antibody. Antibody also one type of protein. And apart from antibody, what another? Your plasma membrane, right? Your plasma membrane, you also containing protein channel, right? Core protein, carrier protein, all that. And also another one that you already learned. Hormone. Protein also acts as a hormone. So the function of the protein is very large. So that is why that protein is very important. One of the component is very important to the body. to the body. Okay, so this one is all the 20 types of amino acid that you need to know it. Yeah. According to MPN syllabus, you should memorize all. You should memorize all. But I think you are human, right? No one can memorize. Even me too, I cannot memorize. Even me, I cannot memorize too. I only can memorize one this glycine only, because glycine is the most basic one. It is the most basic one. But I only can tell you that you can choose any type of the amino acid. Why I say you need to memorize all? Because one of the year MBM, they coming out in structure question, they just coming out this kind of structure, and then you need to name it. Any type, or if no mistaken, 3 type. They coming out glycine, alanine, and another one is I already forget. Any 3 type, they give the diagram lah, these 3 diagram, and then they say PQR. You need to know lah what is P, what is Q, what is R. And then entire question is totally based on these 3 amino acid. So if you totally don't know what is that, what are the amino acid, you totally cannot score that structure question. So yeah, so that is mean that in MPN syllabus you need to know all. You should know all lah, but yeah, you all so I cannot in real world I cannot ask you to memorize all lah. This is unlogic. I memorize all, but try as much as you can lah. Okay. So yeah, as I mentioned, there are only 2 also essential, also non-essential. So you know that essential, there are 9 essential amino acid and also 11 non-essential amino acid. So what are the 9 amino acid? Essential amino acid, these 9 lah. And also what are the 11? This 11 lah. So the main difference is that essential is you must obtain from diet. Your body cannot produce one. But non-essential, yeah, your body can produce this. Okay. So this one is a function lah. As I mentioned, structural function, and also contractical function, storage. Protein has also storage, and also defenses, as I mentioned, antibody, transport. One of the transport, hemoglobin, messenger, hormone, and also enzyme. All that. So you can see that actually protein has the very large range of the function. Okay, so backbone of the amino acid. So as I mentioned before, backbone is R group. So as I mentioned, there are 20 types of backbone in amino acid lah. It contribute to 20 types of amino acid. Okay, so this one is the central carbon. You can see this is the central carbon. It is located at the center, and there are carboxyl group, carboxylic acid group, and carboxyl group, hydrogen atom, R group, and also amino group are bonded to each other to form the alpha carbon. So normally this one is recalled as alpha carbon lah. Since amino acid is at the center. And amino group, as I mentioned, is a H2 lah. And carboxyl group is this COOH, C terminal. When you talk about the N terminal and C terminal, N terminal is the amino group, and C terminal is the carboxyl group of the amino acid. Another one, this one is very important. This is what we call as deuterion. This one is that when the amino acid is inside the water, when it is immersed in the water, soluble in water, I can say when amino acid is in that water, they will form deuterion structure, which is that they will accept and also receive electron all together. Because they containing amino group and also carboxyl group. We know that amino is basic, acid and base. You still remember your SPM chemistry. This one is more focused on chemistry part. You still remember about your SPM chemistry. We know that in acid and base reaction, acid is hydrogen ion, right? Base is hydroxyl group. While the hydroxyl OH, And you know that amino, ammonia, all that is basic. And carboxylic acid, carboxylic acid, any acid is acid lah. Right. Right. So if you still remember, acid is release electron, release proton. Release proton, release hydrogen. And another one is basic, accept proton. They accept proton. They can accept hydrogen ion. One of the theory. This one is chemistry theory, the basic chemistry theory for the acid and base. Acid and base. So in amino acid, due to it containing both basic group and also acidic group, so that they can form internal transfer of a hydrogen ion from the carboxyl group to the nitrogen amino group. So it is very tricky, meaning that where it is released water, where it is in the water, acid substance in zero situation, acid substance they will release hydrogen ion. So as your carboxyl group must release hydrogen ion. Okay, and then your amino group in zero situation, if they are only containing amino group or basic group, when they is immersed in water, they need to receive hydrogen ion. So this one will create a balance that your hydrogen ion for the carboxyl group will go to the amino group, so that they will both go to each other. And then we know that when acid that release the carboxylic acid that release hydrogen ion, they will form carboxyl group, negatively charged, negatively charged, and amino they will form ammonium ion which is positively charged. And this type of structure is recalled as deuterium. And due to this structure, they are also can be act as the buffer solution. Amino acid can be act as a buffer, so that when you add little acid or base is added, the pH of the solution will remain unchanged. This is the function of the buffer. Later in chemistry, I just want to let you know, in your chemistry, chapter 6, equilibrium, you will learn about more acid and base, acidic reaction and also basic reaction. When you learn about that, you will know the function of the deuterium. Maybe now you are confused again, but this is very simple theory only. But in chemistry, they are more complicated lah. In chemistry, you are also learning about the buffer too. They are the definition of the buffer in chemistry. So actually, the concept is still same, totally same. But in bio, I just explain it simple only. But in chemistry, your chemistry teacher will explain more detail lah. But in buffer, when you recall as buffer, meaning that when you add little acid or little base, meaning that it's weak acid or weak base is added, the pH is still remain unchanged. There are no pH change one. The pH is still. If you consider to the normal situation, if you add water I consider as water. If you add acid, whatever acidic, whatever weak acid or strong acid, you add to it into water. When you measure the pH value is will be down. It will directly decrease one. But in this protein, in amino acid solution, when you add only weak acid or weak base, you add to it, your pH still remain unchanged one. So that is why that we call as deuterium. So this is very important, very important. I can say very important structure. One of the major property in the protein. Okay, so what is R group? So R group is a side chain. It is a side chain. So normally your R group is determine the characteristic of your amino acid. So amino acid not only classified into essential and non-essential amino acid. The classification of amino acid is based on the R group. Now different R group will be provide different characteristic to that amino acid. If you consider this basic structure of the amino acid, it is actually act as the neutral. This one is neutral, meaning that pH is equal to 7. if you ignore the side chain, if you assume that the side chain, the R is HR glycine, if you assume this amino acid is glycine, yeah, it is neutral, pH equal to 7. But different side chain will provide different property to this amino acid. So, okay, so there are four main okay, although amino acid can be classified in various ways, one common approach is to classify according to what the functional group of the side chain at neutral pH, either meaning that at pH equal to 7, either your amino acid is non-polar, polar but uncharged, polar, uncharged, or meaning neutral, or negatively charged or positively charged. So you see this diagram, you can see there are total 1, 2, 3, 4, 5. Five main group of the amino acid: non-polar amino acid, polar amino acid, aromatic amino acid, positively charged amino acid, and also negatively charged amino acid. These 5, you need to know it. You need to know it. Now we will go to one by one. So as I mentioned, amino acid normally is considered 4 categories: acidic amino acid, basic amino acid, polar amino acid, and non-polar amino acid. Very important. Acidic is positively charged, acid is negatively charged, basic is positively charged, polar and also non-polar. These are very important. You need to know it, and you also need to know the amino acid too. So wow, the difference is we want to compare the difference between basic and also acid. Acid is based on the side chain. So you can see acidic amino acid, you can see that, wow, the example is these two lah, aspartic acid and glutamic acid. Already mentioned lah, acid. Well, why? Due to the side chain, they also containing the carboxyl group at here. You can see they are also containing carboxyl group at here. So when they dissolve in water, they will, as I mentioned, in acid condition, acid solution, if it is acid, they will release hydrogen ion. They release hydrogen ion so that they will produce acidic solution. So that is why that in neutral, meaning that when you immerse in water lah, pH equal to 7, this amino acid will cause your solution become acid. Your pH will drop. Because why? They're containing 2 negatively charged. One is the basic unit one, another one is based from the side chain. So acid producing effect will be produced. So this one is we call as acidic amino acid. They will produce acid solution. And this one is basic, meaning that they're containing ammonia group at here. So arginine, lysine, and histidine, both 3 of them are basic amino acid where they containing amino group. Normally they containing nitrogen atom. You can see when they are containing many nitrogen atom, you can see they containing ammonia group, amino group, ammonia, and even this aromatic one, they containing high nitrogen atom. All these will provide basic amino acid, meaning that they will when they immerse in water in neutral pH, they need to accept hydrogen ion. So when they accept hydrogen ion, so that they will produce basic amino acid. But as I mentioned, you need to be alert. Well, amide group like amide group CO and H2, so normally asparagine and also glutamine are not considered as basic amino acid. So you can see this. Glutamine. Glutamine. You can see this glutamine. Glutamine are not considered as the basic amino acid. Another one is what? Arginine, lysine, histidine. Yeah, this one is positively charged. Another one, this asparagine and also glutamine. They are polar, but they are not basic. Why? Because they are amide group, C double bond O and H2. This one is not considered as basic. They cannot accept hydrogen ion 1 in simple, so that this one is not considered as positively charged. They are polar amino acid. Okay. Okay, so this one will cause the amino acid, this property, the deuterious property, deuterium property will cause that either your amino acid is in acidic or basic solution at the similar situation, meaning that at pH equal to 7 lah. So meaning that when you are equal to pH equal to 7, they will form deuterium. But if you increase your hydrogen ion, you increase your hydrogen ion or decrease your hydrogen ion, so you can see that at high pH, meaning that your hydrogen ion is very low, deprotonation will be happen. Deprotonation meaning that they will only release the hydrogen ion only. And if your acidic condition is high, your surrounding lah, if your pH value initial pH value is very low, is highly acidic, meaning that protonation only happen. COOH is still maintained. The hydrogen ion will not be released, but your amino group still accept the hydrogen. That is why we call as protonation. Deprotonation is release hydrogen ion. So this one is the concept of equilibrium later that you will learn in chemistry lah. Okay, so this is the protonation lah. So normally when your hydrogen ion is very high, meaning that your pH is dropped, so that your carboxyl group only will accept the proton to become uncharged COOH. So your overall will be the positively charged. So compared to deprotonation, where your hydrogen ion is very low, pH is high, meaning that in very acidic condition, so that your carboxyl group will lose the proton to become CO negative, that your overall will become negatively charged. And then this one is the differences between polar and also non-polar amino acid. So normally polar amino acid normally is hydrophilic, they can soluble in water, but non-polar amino acid they cannot soluble in water. This one right, polar can soluble, non-polar cannot soluble. This is a general concept lah. And also normally polar amino acid, your R side chain normally is partial positive, either partial positive or either partial negative, so that charge group. But in non-polar, normally they are neutral, totally neutral. Normally they are neutral. There are no charge group at there. No charge group. Does not carry any charge group. So that polar amino acid normally can found on the surface of the protein, but this one is found interior part of your protein. You know protein. I already mentioned protein is the combination of many of amino acid. So polar amino acid you can find at the surface, and non-polar is found on the interior part. And normally polar amino acid you can soluble in aqueous solution lah, like water, but non-polar is soluble in organic solution like alcohol. So these all the polar amino acid, including tyrosine, serine. You can see they are hydroxyl group 1. Hydroxyl group, they are polar group 1. Hydroxyl group, and they got carbonyl group, amide group, and also sulfur group at here. Okay, and then you can, if you see the non-polar, normally is totally hydrocarbon chain 1. Like glycine is the basic unit lah. Glycine and alanine you should remember lah. This one is most easy one lah. Glycine is the general one, and alanine is only containing one methyl chain. Valine is isomethyl, and then leucine, isoleucine, proline, tryptophan, phenylalanine, also methionine. All these are considered as non-polar amino acid. You can see that they are normally is containing hydrocarbon chain. Okay, so that's all about the amino acid. Okay, next we go through the peptide bond. Okay, so we already learn about the amino acid. Now we go through the peptide bond. So what is peptide bond? So peptide bond is the amide type of covalent bond that link two consecutive alpha amino acid from C1 of one amino acid and N2 of another. So if I see you the diagram, more easy like this. So these two are glycine, this glycine 1 and glycine 2. So if you want to form peptide bond, one part of the hydroxyl group and another part of the another amino acid of the amino group will release one hydrogen molecule, so that OH group, hydroxyl group from the carboxylic acid, and also one hydrogen atom from the amino group will be released out to form water molecule, so that this is condensation process. They will form peptide bond. So they are the amide covalent bond at here, CN. The C bonded with N. So this C is one amino acid, one glycine, and bonded with another glycine. So this one is peptide bond through the condensation process, where we can release water, release water, yeah. Okay, so dipeptide is the structure. If there are two amino acids joined together, then this structure we call as dipeptide lah. Okay. And then this one is of the important. So when there are many condensation process, if condensation process is rapidly occur, so they will form the protein molecule with different structural level. So either they form primary structure, secondary structure, tertiary structure, or quaternary structure. So if at the higher level of structure, the more complicated of the protein structure, and also the higher number of amino acid. So in primary structure, very simple. You see this diagram. Primary structure is just a single polypeptide chain. Polypeptide chain is that when there are many amino acids are bonded together through the condensation process, they are bonded together, they will form primary structure, which is there are only single polypeptide chain only, like this. Only single polypeptide chain. So normally this one is the main structure is covalent bond. They are only covalent bonded at here. So you see this one of the example, insulin lah. There are only single polypeptide chain only. Okay. Another one, secondary structure. Okay, so this one, secondary structure, is that there are one bond which we call as hydrogen bond. Yeah, that you already learned in chapter 1. But this hydrogen bond is not formed in water. This hydrogen bond is formed if within the same polypeptide chain, there are hydrogen bond can form. If you still remember, in water, how hydrogen bond can form? Partially negative. If there are the one water molecule and bonded to different water molecule through the partially positive hydrogen and partially negative oxygen, right? And different water molecule, then they join together, they will form hydrogen bond. Similar to the protein too, but in protein they are not different protein lah. They're just that different location of amino acid. In the different location of the amino acid, if there are partial negative oxygen and also partial negative hydrogen, they're able to form hydrogen bond so that they can coil or fold to the polypeptide chain, so that you can see the polypeptide chain is like that. Better than this one is just a basic unit only lah. You just join like that only. But this one is not join. They not only join, but the structure is coiled to each other. They form coil structure. You can see they switch, right? Macam spring macam ni, and you can see ini macam kertas. This is what we call as folded. So there are two common type of secondary structure. Either is alpha helix is like that. This one is the helix structure, coiling structure like that, or beta pleated sheet structure. This one like that, like a paper. You can see it is like a paper one. But both of them are held by hydrogen bond. Why they can form like that? Due to hydrogen bond. Hydrogen bond can make the structure like that. The key component is hydrogen bond. If no hydrogen bond, they will just form like that only, like this only. Just a covalent bond, they join together, only like that. But this one, due to the presence of hydrogen bond, so that they will form helix structure and also beta pleated sheet structure. So this one is how alpha helix structure. So it is a right-hander helical structure, and each helix contain 3.6 amino acid residue. This one is average. So normally your C double bond or group of each amino acid is hydrogen bonded with NH group, meaning amino group of another amino acid, which is situated 4 amino acid at here. So meaning that you see here, when there are the amino acid at here, and there are another amino acid here, so that they can form hydrogen bond at here. The one of the O group and also one of the N group is can form hydrogen bond at here. So like a shortcut lah. They will not only bonded like that, they will form a shortcut like that, so they can join together, tied together. Dia macam tarik dia ke sana, so that they conform the helix structure like this. So this one you can project outward from the twisted backbone. A beta plated sheet is totally fully extended, meaning that beta strands line up side by side, forming beta plated sheet, and it is stable between also hydrogen bond. So you can see it like this, just side by side, like this. Like this, can run parallel or anti-parallel to each other. R group are alternatively projected to above and below the plane of the beta sheet. So you can see this diagram. This diagram is like compared to the alpha helix. Alpha helix is like a single polypeptide chain, and then they just squeeze at this, like this. Macam ular lah macam tu. But ini macam paper dia. Tegak-tegak, all the amino acid is straight away, and then just that they can form the macam join macam ni, and to form the hydrogen bond parallel, anti-parallel or parallel to each other like that lah. Okay, so this is the summary of the differences. You can read yourself. Okay, tertiary structure is more complicated. Because this is 3D structure already. Compared to primary and secondary, both of them are still 2D. Like a paper only. Like a paper you see. Normally it's like a paper you see only. But this one is 3D structure. Tertiary is the 3D structure already. 3D structure already. But still, only one polypeptide chain form. Primary, secondary, and tertiary, 3 of them are only one polypeptide. There are no 2 polypeptide, 3 polypeptide. Only one polypeptide. So normally it's still similar to secondary structure. What happened in secondary structure, tertiary structure pun still happen, but there are additional interaction occur between polar, non-polar, acidic, and basic R group within the polypeptide chain to contribute the 3D shape. So you see that there are total 4 interaction. Hydrogen bond is one, compared similar to secondary structure. Another one is disulfide bridge, hydrophobic interaction, and also ionic bond. These 3, they can form these 3, another 3. So disulfide bridge, as you see that in amino acid, certain amino acid they containing sulfur. Right, in the side chain, you still remember the polar amino acid and also non-polar one. Both of them, some amino acid can containing sulfur one. One of the example is methionine lah. Methionine containing sulfur. Okay, so they can form the disulfide bridge to each other. This one is one interaction, and another is hydrophobic interaction between the non-polar and non-polar group. You can see this. This one, if not mistaken, is valine, right? Valine. Right, valine. If not mistaken, valine. Yeah. This one is the hydrocarbon chain. When they are hydrocarbon chain to each other, they can form hydrophobic interaction like this. This one we call as hydrophobic. Why hydrophobic? Both of them are hydrophobic. So they can form the weak interaction like Van der Waals force. For very weak Van der Waals force to form we call as hydrophobic interaction. And another one is ionic bond. They can form also ionic bond at here. If they are the different charge, If they are the if they are the positively charged and also negatively charged amino acid, the side chain, not all of them are side chain. You can see all of them are the side chain. Where the side chain of the amino acid is either positive or negative to each other, they can form ionic bond. So you see the interaction between an ion and cation of the R side in the polylipid. Hydrophobic is when there are two nonpolar to form. A hydrogen bond, then there are the hydrogen atom from one R side and another one is another R side. A disulfide bond or disulfide bridge is when there are the sister amino acid that can form SH group, SS bond, sulfur bond. So these are 4 interactions contribute to let it can form 3D structure. And last one is quaternary structure. Okay, quaternary structure is also 3D. There is also 3D, but highly complicated. Why? Because it is containing made up of many polypeptide chain. Compared to tertiary only one polypeptide. One polypeptide they already can form 3D, but this one is containing 2 or 3 or 4, at least 2 polypeptide, so that they can form we call as quaternary structure. And then the subunit may be same or may be different, meaning that the polypeptide chain can be totally same, meaning that A chain and B chain, the sequence of the amino acid are totally same, or they can differ, meaning that the A chain and B chain are totally different, or there are something different at there. Maybe A chain containing 100 amino acid, B chain containing only 60 amino acid, like that lah. And then all the interaction in the tertiary structure still happen in quaternary structure. The difference is that they contain more than one polypeptide chains. So one of the major example is hemoglobin. So you can see this structure, this hemoglobin. Hemoglobin containing 4 polypeptide chains, 2 alpha chain and also 2 beta chains. Beta chain, 2 alpha and 2 beta. So you can see these, the lower alpha, the upper one beta lah. Okay, hemoglobin is one of the famous example lah. Okay, next we go through the denaturation and also renaturation. Okay, we already cover all the basic of the amino protein, the level structure. Okay, now if we know that protein, if you want to break down is very easy. If you want to alter the shape of the protein, it's very easy. It is not just that hydrolyze. Hydrolysis is just if you want to break down peptide bond. But if you want to break down peptide bond, it's not easy. Breaking up the peptide bond is not easy. But if you want to break down the 3D structure of the protein, like this 3D structure of this, or you can want to break down 3D structure like this, it's very easy. Why? You need to know that the interaction, all these interactions are very big interaction only. Compared to a covalent bond, at here the covalent bond, the peptide bond between the amino acid, this one is strong. If you want to break down it, it's still hard, it's quite hard. But if you want to break down, especially hydrophobic interaction, disulfide bridge, hydrogen bond, all these 3, very easy. If you only your environment change, like your temperature is too high or your temperature is too low, or you add the acidic condition, or you add a lower acidic, or you go to radioactive environment, your amino acid, your 3D structure of your protein will change, very easily to change. So this one is that we know as denaturation and renaturation. Denaturation is your protein is already denatured, meaning that your protein is altered in shape. Altered in shape meaning that your function of the protein is different, already interrupt. You already interrupt your protein function. Like quaternary, we know that there are 4 chains. If they are bonded together, but if there are something happen, when the chain is break down, or you already break down any interaction in this, maybe already from 2D, 3D become 2D, like that, your hemoglobin cannot function already. This one we call as denaturation, and renaturation is reforming lah. But actually in real situation, you want to reform again the protein is not very easy compared to denature. If you want to macam senang kan, nak rosakkan sesuatu benda senang je. Nak rosakkan just pukul je, buang je, rosak. But nak repair susah kan. Macam accident lah, accident langgar je, okay lah satu saat. Tapi awak nak repair your car, wow, you need to take time. So similar to protein too. You want to break down the protein structure very easy. It's just very easy. But you want to reform your protein again, very difficult. Very difficult. Okay, so normally denaturation, change in specific 3-dimension structure, especially in tertiary and quaternary protein. Normally in our real situation, especially in our body, majority of our body protein is tertiary or quaternary one. There are very less is primary and secondary. Very rare. Primary and secondary normally is in the prokaryotic bacteria lah, or very small organism one. Like human complex organism or bigger big animal like that, normally our protein is tertiary or quaternary. You see human hemoglobin is very important. If your hemoglobin is breakdown, is already broken, you can imagine the function lah. Okay, so the 3D structure of the protein is relative unstable. I can say unstable. Normally these two, although ionic bond, ionic bond is strong, right? Ionic bond you see strong, but this ionic bond is fat ionic bond, not ionic bond that you learn in chemistry. You know that ionic bond in chemistry is metallic bond. It is between the formation of the bond between the metal and metal one, or metal non-metal. That one is ionic bond. That is very strong. But this one is just a fat bond only. Because this one what you call as ionic bond, because of different charge only. Different charge, then you call as ionic bond. But this bond, if you compare to the chemistry, the real one, the metal bond, actually is still weak. It is still weak. So you don't want to confuse with the ionic bond in this and also chemistry, that you learn in SPM or in your chemistry. This ionic bond is fat ionic bond. And denaturation of enzyme, antibody and protein hormone will cause your function loss. Although your amino acid sequence still same, they will not break down your amino acid sequence, they will not hydrolyze your amino acid, your peptide bond, but your peptide bond still remain. But your interactions change, your 3D structure change, your function also change. Like quaternary, like your hemoglobin. Hemoglobin, you know that play the role in carry oxygen, right? Carry oxygen also carry carbon dioxide. If your hemoglobin is breakdown, how your blood, how do you carry oxygen? You cannot carry oxygen already, then you die. So it is very important. So what can cause denaturation? This either heat or radiation. When you heat it, heat your protein, so that is why that at very high situation, that is why human cannot survive at the high temperature surface, high temperature places at the long time, because not only you cannot tahan, not only hot, but your enzyme also cannot tahan. Your protein, your internal body, including enzyme, antibody, all that also cannot tahan. Because this one, in very hot situation, you will increase the kinetic energy inside the atom, between your atom, so that your protein, your interaction between protein will be very active. If too active, hyperactive, it can break down your hydrogen bond and also ionic bond one. When it break down, 3D structure after. And another one, strong acid and strong alkali. If your protein is exposed to strong acid or strong alkali, what happen? Additional of hydrogen ion, this one is strong acid. If strong acid is added, it will combine with carboxyl group, COO negative in amino acid to form COOH. So reduce hydrogen ion in alkali cause your ammonium group in amino acid to release hydrogen ion to alkali medium to form amino. So this one will cause your ionic bond is break. You know that if you want to form ionic bond is you need to have a positively charged for amino or ammonium, and also negatively charged from carboxyl, right? Then you can form ionic bond. If not, if it like that, already form neutral, already form COH back, already form NH2 back, so no charge, so already form neutral, so your ionic bond are broken lah. There is highly stock concentration or heavy metal also can cause. So normally this one is highly stock concentration due to you can form very electropositive ion, form a strong bond with carboxyl group in amino acid. Meaning that this one is according to ECS, electrochemical series. This one, if they are highly electropositive, so normally is metal ion. If they are metal ion, like sodium ion sodium ion, you know that sodium ion, potassium ion, all these, potassium ion, magnesium ion, zinc ion, copper ion, all these have highly reactive compared to carboxyl, compared to hydrogen ion, right? Magnesium ion, sodium ion, copper, or even zinc, all that is highly electropositive, highly reactive compared to hydrogen ion one, so that they will form bond instead with the amino group. Actually, in real situation, your this COO negative, your carboxyl group should form bond with the amino group, right? In natural situation, but due to they are highly electropositive ion, highly reactive, so that your carboxyl group will not bond with the amino group. They will form with the any electropositive, so the ion, magnesium ion, zinc ion, all that lah. Similar negative. If they are highly electronegative, they will not bond with CO negative. They will bond with other, so like hydroxyl group or other lah. Also, organic solvent and also detergent will disrupt your hydrophobic interaction. So this is the cause of denaturation can be occur in your in protein. Okay, if you want to renature your protein, so normally it will sometimes spontaneously refer to its original structure. So normally this one is you can provide physical and chemical environment to normal with the denatured agent removed. So protein functional structure remain the same and normal biological can be carried out. Okay, final, we go through the classification of protein. Okay, this is the final. But this one you don't want to confuse with the level structure. There's not primary, secondary, tertiary, and quaternary. These are no classification. That one is no classification. That one is the level of structure. If the exam want you to classify protein is this topic, if exam ask you to describe the classification of protein, you write primary structure, secondary structure, all that is no classification. That one is level of structure. You need to different it. You cannot confuse it. You confuse it, then too smart. Okay, so normally in classification we are two main classification. Either you are simple protein or conjugated protein. Simple protein is that your protein only containing amino acid, but conjugated protein is contain not only amino acid, they are also containing what we call as prostatic group, a non-protein material, any non-protein material. One of the example is hemoglobin. Hemoglobin, we know, right? They contain hem group, meaning that this one is metal ion. We know that it containing metal ion, the ferrum ion at there, fetal atom in the hemoglobin. So hem, your metal, your ferrum is prostatic group. So they are not only containing amino acid, they are also containing the ferrum. So hemoglobin. So these are all the examples. Also glycoprotein. Glycoprotein also one of the protein. They are not only the glycoprotein, right? Glyco meaning that they are carbohydrate chain bonded to that protein. So this one, your prostatic group is carbohydrate. And also nuclear protein, casein, or this is example. Another different classification is globular protein and also fibrous protein. So you can see that globular protein normally is the very circular shape, 3D shape, circular 3D spherical round structure. So normally it is made up of any type of protein structure of the protein amino acid can be formed like that. So normally this one is the explanation. And fibrous protein, if you see that fibrous protein is a long and narrow and normally repeating set of amino acid. So normally the role is strength and support. Strength and support. Just now, this one is role as enzyme and transport. Strength and support. And production is movement, and also surface is covered in non-polar amino acid. Okay, so this is the difference between fibers and globular protein. Okay. And then this one is just a general function of the protein. So this one you can read yourself lah. I don't want to talk much. So yeah, function of protein, as I mentioned, function. One of the function is enzyme lah. So this one is one of the chapter 4. You will learn it. So I don't want to talk much about this. So chapter 4 I will talk more detail lah. Another one, as we mentioned, is we can provide protection and also support of the structure. So normally like collagen, your elastin, fibronin, all this is protect or support to your body. And movement, like your actin, tubulin, myosin, cytoskeleton, all these also involved in movement. And defense, I already mentioned antibody lah. One of the antibody. Not only antibody like fibrinogen and thrombin that play the role in blood clotting. Another one, regulation, as I mentioned about hormone. This one is growth hormone. And blood glucose layer, as I mentioned, insulin and also glucagon. This one you will learn more detail in semester 2. This one is ceruloplasmin. This one you don't need to know lah. This one is more about biomedical part lah. Okay. And final one is storage. So normally you can protein is also act as the storage. You can provide the energy lah. You can also provide energy to the human. And these are all the function overview of the protein function that I extract from the Campbell. You can read yourself. So I think that's all for today lah. That's all for this topic.